Hello one and all, myself Dr. Pankaj Gautam. I welcome you to my laboratory where we carried out all the experiments related to this patent which has been granted to us recently. And I would like to first introduce my team. To my right hand, Dr. Ashish Thaplyal. He is a renowned cell biologist and neuroscientist. And then we have Dr. Naveen Kumar. He is a very famous molecular biologist, and his area of research is fungus, and he is working on candida also. And then I have with me Dr. Nishant Rai. He is a renowned microbiologist, and his area of research was virology. So the story began in 2014 when we filed our patent, but those stories started even before 2014 when we were working on a project related to the diagnostics and development of monoclonal antibody. In that project, this project was funded by USPD, the state funding agency in biotechnology. And at that time we got this idea, and then we all sat together, we put our head together, and we developed this concept that why not we can detect this pathogen by using the PCR. So PCR, it is called the polymerase chain reaction. It's a kind of molecular xeroxing through which we can amplify any gene. So in our case, we are trying to amplify gene of a related pathogen which is causing the disease, that is Salmonella Typhi. This pathogen is responsible for the development of the disease called typhoid. So this is the history and genesis of our experiments, and then started a long chain of hard work where we conducted a lot of experiments in the laboratories and we accumulated data. After accumulation of the data, we filed our patent and we provided all the required documents and evidences. And then we also tested this in samples we obtained from various places, and we found very promising results. Then we went in for the PCTs at international level, and PCTs, it's a kind of patent cooperation treaty which is signed by more than 100 countries of the world, and they will evaluate and examine all the new inventions and their potential at international level, and then they will pass their opinion about the commerciality and inventiveness and novelty of that particular invention. So we received very encouraging response from international community, and after that we waited patiently for last 6 years, I would say, and this was a time when we faced a lot of criticism also. Sometime these comments were very nasty, they were not very pleasant at all, and we were planning somehow that our invention, definitely it's working, and we can prove it to the world that we can detect Salmonella from any sample satisfactorily and with a lot of promises. Earlier, as we know, there are several tests for typhoid like Vidal and Typhidot. These tests are cost effective, but they are not promising. If you compare them with a PCR based or DNA based detection system, then you will find that it is more convincing and reliable. Even nowadays, as you can see, that the reliable test is considered for COVID-19 is RT-PCR, and it's not the antibody based test, even though it's quick and rapid, but the reliability lies with the RT-PCR. So thus you can judge the potential of PCR in the diagnostics, and we have to put in a considerable amount of effort and hard work in proving our novelty and commerciality, and finally we got the reward in terms of the patent grant, which was definitely phenomenal, I would say. And at this point, I would like to emphasize the support from the management, especially from our Honorable Chairman, Sir, Professor Dr. Kamal Ganshalaji, who was very encouraging and supportive, and his motivation was very, very instrumental factor for us that we moved this far and we have achieved this feat. So management has offered a lot of help and support in terms of seed money, in terms of research facilities, in terms of research awards, and very, very much encouraging from every post from every person received this encouragement. Our Vice Chancellor, I would like to thank our Vice Chancellor also, our Pro Vice Chancellor, even our patent attorney was very instrumental in helping us, and I thought it's a wonderful day for all of us, and we feel proud today on this achievement. And it is the blessing of all of you, I would say, that we have achieved this far. Thank you.
Hello everyone. As my colleague Dr. Pankaj Gautam has already briefed about the invention that has recently granted through Indian Patent Attorney Office. Being a molecular biologist, my contribution basically in this invention was, because this invention depends upon the detection of the DNA of the pathogen. So how we detect that DNA of the pathogen? We take the sample and we isolate the total DNA from the sample. After taking the blood sample from the serum, the total DNA is isolated. Then in this invention we have one specific primers, that is those primers are novel for this invention. So those primers they get attached to a particular region of the pathogen's DNA, and after this the primers and the genomic DNA sample isolated from the blood, they are mixed together in a tube that is called the PCR tubes. These are the 200 microliter tubes where we set the reaction, and inside these tubes we put the primers, template, and the enzymes, buffers, some other components. And then this is the instruments, my dear friends, where we perform the PCR reaction. PCR means polymerase chain reaction. As sir has already told you that this reaction makes the Xerox copy of the DNA. So the DNA is amplified in this reaction, and for your information, if there are very feeble amount of the DNA, even there are the template, just 10 or 100 copies of the template, then this instrument is going to make it to million in just 2 hours. So we set the PCR reaction here. We set the tubes. After setting the reaction inside the tube, we put the tubes here in the slots provided here, and then we run the PCR. And after running the PCR, after 2 hours or 2.5 hours, the DNA is isolated. That DNA is already that is detected through other systems. There are some other assembly and instruments which are used for the visualization of the DNA. Thank you.
Hello everyone, I am a microbiologist, and it was a wonderful experience with all the inventors with me. So I was supposed to work on Salmonella Typhi, and I was involved in that cultivation, identification of Salmonella, and all the microbiology laboratory works. So we could identify the DNA, and for the information of young researchers and students, this is gel documentation system, and we keep the agarose gel here and visualize under UV light. So this camera actually captures the images and displays here. So we could identify the specific bands, right? And it was a novel site which was amplified. So it was a wonderful experience, and we have a research policy here, and the management supports in terms of the seed money grant, then they support the instrument which is required in the particular patent, and we could work in the team, so it was a successful achievement. So with this I would like to give you a message that this is the platform where you can transform your dreams into reality. Thank you.
I'm going to talk about a few things about which are very important in research. What is a patent and why are we talking about it? Patent is something that is a novel thing that has been developed and that was not there before. So this patent is about detecting Salmonella, and earlier everyone has heard about this Vidal test. Now this new test that has been developed, this is a new technology. The details of the technology has been told by my colleagues. The very important thing that I wanted to share with you is that these are three fundamental things. Always believe in your research, the first thing. no matter what happens, keep doing your work. Second thing is always believe in your team. Always have a very, very good teamwork. No matter what job has been given to you, no matter what you did there, even small things, teamwork is really important. And the third thing is you have to understand that keeping in pace with the call of the nation, Atmanirbhar Bharat, and you must have heard so many calls. The achievements of this patent, they are really of international level, and the entire diagnostics facilities, the entire diagnostic scenario is going to be affected by this patent, and that is what is really important about this patent. So in science, it's really important to ask the questions that are the keys. And if you have a very good team, if you've got a very good management to support you, even filing a patent is very, very expensive for a normal researcher. And all these background facilities, they are provided by the university. And this entire setup has led to this very good achievement. And this is truly the motto of the university, transforming dreams into reality. And this is one of a very good example. Thank you.